Hello, everyone. I'm Monica Birkhead and one of the extremely privileged microscopists to have been awarded a travel grant to attend the EOE8 in Stellenbosch and the Facilities Management Workshop in Cape Town. I would like to thank the grant selectors, the organizers, both local and international, and the sponsors for giving me this amazing opportunity. The grantees were asked to introduce themselves with some background to our education, current place of employment, and work activities. So, I'm a South African from the country with which I'm sure you're all more familiar than before the year E8. It's divided into 9 provinces, the smallest of which is Gauteng province, considered to be the economic hub of the country. Gauteng is divided into 5 municipalities, and I have lived in Ekurhuleni for the last 5 decades. I did all my tertiary education at the University of the Witwatersrand in Johannesburg, which was where I used a light microscope for the first time, as the school I had attended was very small and ill-equipped, although I loved every minute of it. From my second year of study, I was mentored by Dr. Millicent Freen, a botanist and microscopist, and in her retirement, a renowned botanical artist. She employed me during vacations whilst tutoring me in histology, plant anatomy, taxonomy, and best of all, she introduced me to electron microscopy. I gravitated towards a taxonomy and ultrastructure of marine phytoplankton, particularly the families comprised of cells with ornate polysaccharide scales and taxonomically revealing flagella apparatus ultrastructure. I've included this hand-drawn reconstruction of the flagella apparatus of one of the species I studied just for your amusement, because it was done using printed micrographs of serial sections. At the time, there was no software or automation available. Following my doctorate, I took a break from academia until I was lured back to electron microscopy as a research assistant in the Faculty of Health Sciences, again at the same university but on a different campus. When the research funding ran out, I moved to an aquatic toxicology testing lab for a privately owned ecological consultancy. When this was bought by an international company, I fled the corporate world with its endless meetings and delegations, and I moved to my current job as an electron microscopist at the National Institute for Communicable Diseases, or the NICD. The NICD is a division of the Parastatal National Health Laboratory Service, and it shares a large campus with the corporate offices of the NHLS, which is situated on the border between 2 of the Gauteng municipalities. The NICD is divided into a number of centers which reflect the spectrum of syndromic communicable diseases, as well as housing the National Cancer Registry. The Center for Emerging Zoonotic and Parasitic Diseases has 6 working groups and includes BSL-3 and BSL-4 laboratories. The EM lab is one of these groups, but it also acts as a core facility for other NICD centers, which I've highlighted in pale green, as well as for other NHLS staff. I have a 15-year-old 120 kV Technite 1210, which is kept functional by Mohammed Kohler and Gunther Meyer, and I'm supported and encouraged by the head of the center, Dr. Jacqueline Weyer. These other names are those of my friends and current colleagues for whose help and interaction I am very grateful. As a core facility, I'm immensely lucky to work on such a variety of pathogens, and I'm constantly in awe of their exquisite ultrastructure. These are just a few diagnostic and research specimens of interest. Cultured Ebola virus for use as a positive control for molecular diagnostics, a Tanapox virion from the first clinical case to be reported globally in 17 years, which we did last year, a Parapox virus particle from a lesion on a farmer's hand, sectioned material from a cultured isolate of a representative of the group that includes rabies virus, which are characterized by having bullet-shaped virions, SARS-CoV-2 particles in placental tissue, which was the first published ultrastructural evidence of vertical transmission, part of the kinetosome of the hematophagus protozoan causing African sleeping sickness with its beautiful microtubular cytoskeleton. Streptococcus with the polysaccharide capsule stained with a cationic dye. The capsule is linked to immune evasion and virulence, and vaccines are formulated to target the capsular components. Then we have the incredible layering of an anthrax exospore, and then a fungal pathogen, Cryptococcus, also with a stained capsule. Cryptococcus is the second most common cause of death in people living with HIV. A transverse section through the polar filament, polar lamellae, and polariplast of a mature spore of Microsporidium, which is an obligate parasite as it has no mitochondria of its own, depending entirely on the host for all energy requirements. Perfect parasite. And finally, one of my current projects, which is the diagnosis of primary ciliary dyskinesia. This is a genetic disorder which affects mucociliary clearance of the respiratory tract, and for which ciliary ultrastructural defects can be used to identify the majority of cases. Thank you all very much for your attention.